What's up, everyone? Great to check in once again. I have a little video here because I have a new article up on my Substack I wanted to tell everybody about. It is called The Directed Evolution of the Lyme Disease Spirochete from Borrelia anserina as a Man-Made Event in the Biological Weapons Race. So this is an article it's a really deep, long, it's a real deep dive into um, the connection with the Borrelia burgdorferi, or the Lyme clade, the group of Borrelia that are considered Lyme disease, and Borrelia anserina, the bird Borreliosis. And it was inspired by my favorite critic, Henjen, who he's like this anonymous X account. He's a troll, and he loves to bother everybody. He loves to harass, stalk, just throw feces everywhere, basically in a figurative sense, of course, and just tries to muddy the waters as much as he can, try to smear people as much as he can. He fights really dirty, but yeah. He put this long article together with the help of Grok to try to debunk my book, and he prides himself thinking that he's completely single-handedly demolished the sleeper agent when as nobody else before him had done. He's such a joke. And he thinks that because of by pointing out that he thinks there's no way Lyme disease could have come from Borrelia anserina because of the distance. The cited 25% divergence in the genomes between Borrelia burgdorferi and Borrelia anserina is what he uses to say that there's no way this could have happened. And he puts together this like simple math on small scale point mutations and how many years it would take for Borrelia anserina to become the Lyme clade. And it just totally completely removes the entire story that I laid out. Like it doesn't take into account any of the genetic engineering, the bioengineering that Erich Traub was using, and the factors surrounding it. And I wasn't talking about just small-time laboratory enhancements like you would see in today's work. This was something completely different. And it wasn't just animal passages that was involved in creating these biological weapons. And I wish I had been more clear about that in the sleeper agent, because I know Loftus talked about it in the beginning in the foreword, saying that animal passages can greatly enhance diseases, but it was a lot more complex than that. It was a lot more sophisticated than that, actually, because what he was doing was he was mixing a bunch of infections all into one and then passaging those at the same time. And that's and the fact that all of those create this immune tolerance like disease, or this immune tolerant state, it makes the mutation rates go up significantly, because immunosuppression causes massive rise in mutation rates. And especially when you have a mix of infections like that, it's a completely different story. And then the fact that Borrelia anserina was only used to one host, birds, and one vector, soft-bodied Argas ticks, and probably for millions of or billions of years. And then suddenly he's given it broad spectrum host adaptation and now, because the Lyme clade can infect hundreds of new animals, right? So you're going to go from that to that. You're going to have a crisis situation for the spirochete, pretty much. And it's going to be functioning under a completely different set of rules, which I laid out in this article. It's not going to be functioning by these small scale point mutations, these small mutations, or just mutations elevating somewhat. It goes by a completely different set of rules, and I laid it out in the article, like this SOS, stress response system, or this RPOS. It's called the RPOS. I forget what it stands for, but it's part of the backup plan for when the spirochete enters into a crisis situation and needs to rapidly evolve to survive it. And normally, I would say it probably wouldn't have survived if not for the fact that Erich Traub had given it all of the necessary tools to actually survive and evolve. And especially mixing it with a whole bunch of different infections, being able to induce immune tolerance into each of these hosts as it was starting to adapt. And now, I think a lot of the divergence, or a lot of the genetic distance that you see from Borrelia anserina to the Lyme clade, Borrelia burgdorferi, actually happened once it was released. It may not have been completely, it may not have been so distant when he had just produced when he had just done the bioengineering at Insuleremes. I think once these were released globally, in establishing in different ecosystems and going through these unpredictable set of animal passages, or serial animal passages, alternating between all these different animals in the wild, that's where you see the big divergence occur in these ecosystems. And that's actually what we see, is that you have these European types, you have all of these different types of the Lyme clade that are specific to different continents, even though you also have some that go across different continents, and they infect animals on, that they're pretty steady on. Like Borrelia burgdorferi, for example, has a global reach, and there's some that are just like more common in Europe. There's like, I think it's Borrelia apzelii, and you have some that are common to South America. You have some, and scientists have said, or papers have shown, that the ecosystem can bring a lot of divergence to a pathogen. Like when they set up in certain ecosystems, it can change them considerably. So that's basically where I would say the divergence happens, because I wasn't clear about it in my first article. I was more focused on this the 16S rRNA gene, which is like the most conserved gene, and that's usually what they go by, and that had like I don't know, up to a 4% divergence from the Borrelia anserina, which isn't actually that much, because even within Borrelia anserina itself, it can vary up to 1.3%. And in the Lyme clade itself, there's up to a 2% divergence. So there's, these things, the way that Erich Traub was doing things was a lot different than the type of bioengineering that we see common in papers. It's just, nobody has done what he was doing, where you take a bunch of different infections, roll them all together, and then adapt them to tons of new animal species. And he was very he made a life study of viruses, especially there's some viruses he studied that can infect a bewildering number of different animals. Borna disease virus, for example, can infect so many different animals. And the influenza viruses and LCM virus can also infect a lot of different animals. So that was something that was definitely on his mind, was how can I give this broad spectrum host infectivity? so it can spread freely in an ecosystem. And I basically made this paper in response to my favorite critic, Henjen, and he uses, and the only thing I know about him is what OpenVAET said about him in an exchange in one of his earlier articles. He said, in the following developments and controversies, Henjen, ex-Mongol Phi, 8-bits data mixer masquerading as experienced conspiracy theorist, produced yet another one of these chat GPT analytic flows that he masters like no one else and is urged to demonstrate that the jabs are working as intended. The character appeared on the Twitter scene at the end of 2023 and his only endorsed conspiracy to date is all Jews are bad. Don't ask him to justify this view or that makes you a baby truther, only worthy of his despise. And despite posing as a truther, he viciously defends the COVID vaccine, and he basically doesn't believe in biological warfare. That all the stuff that people are claiming is biological warfare is just natural spillover. And he used to have the flag of the Mongolian, or the flag of Finland on a Mongolian character, and that was his image. And then he changed it when I made that image of him rubbing shit all over himself. You got to put that back. You got to put that back. It's just not the same. The cryptogram thing, the weird little cryptogram thing you put up, just it's not the same. It doesn't have the effect that you used to have with that Mongolian. It's just not the same. You got to put that back up. But yeah, it was he produced this really long article. I mean, really long. Most of it just smear tactics, and most of it just attacks John, and a lot of it attacks John Loftus. And I don't see eye to eye with John Loftus on a lot of things, but that doesn't mean that some of what he says can't be useful. And what I was doing, since he was the first one to publicly say that Lyme disease was connected to biological warfare, I reached out to him and I started asking him questions. Then he introduced me to the source who actually was the original source for that information, which I can't say his name, but I did meet him and I got a lot of good information from him, and a lot of that information helped me write this book. And I just, based on that, I had John Loftus write the introduction because he's somebody who was in government. He's somebody who had a high-level security clearance, a Q-level clearance, and so did this other guy who's the biodefense source. And so I asked him to write the Floyd, and it's like, I don't see eye to eye with him on everything, and he sees, like, he definitely sees things very differently than I do. Okay, let's just put it that way. But, Henjen just tries to make connections. He tries to link him to all these people that he has. Like, he tried linking Loftus to secret societies and stuff. And it's like, Loftus is the most unspiritual person you will ever meet in your life. He does not believe in God. He does not believe in spirits. He does not believe in anything occult. It's all nonsense to him. So I thought that was pretty funny, where Henjen was saying that stuff about him being connected to Freemasons and stuff. And he even mentioned Michael Aquino at one point, just because John Loftus was a military intelligence guy and had known this other guy who might have known Aquino. Aquino, though, Loftus did a lot of work on defending the Jews. Michael Aquino, and Loftus was very against Nazism. Michael Aquino loved Nazism. He actually, I'm pretty sure he wore Nazi regalia at one point, like, I don't know, or he had it or collects it, and he went to Germany. He was really into the Nazis, Michael Aquino was. So I was like, those two would not see eye to eye on anything, and those two would not be friends. He would have laughed at Aquino. He would have thought Aquino was a complete joke, looking like, one of the monsters or whatever, with the weird eyebrows and, all dressed up in a satanic and, monk suit or something. Like, Loftus is not spiritual whatsoever. He's not, He's not, he's a complete atheist and sees things in the scientific. Actually, he would probably see things more like Henjen, because Henjen likes the establishment view of everything and loves vaccines. Well, so does Loftus. And, I wish I could have gotten through to him about that stuff. And it's just, some people just don't want to see the truth. That's the way I see it. I showed him so much stuff, showing him how harmful those jabs were, and he just does not want to see it. I mean, I think he does get it a little bit, but I think, like he thinks the whole, well, I won't get into that, but let's just put it this way. We don't see eye to eye on a lot of things, but that doesn't mean that we couldn't come together on some certain things and work together to try to do something better for the world at large, because I believe in um unifying with other people regardless of who they are, if there is good that can come of it. um so most of Henjen, I linked to, Henjen complained that I didn't, in my other thing, I didn't link to his article. He thought I didn't have the courage to link to it and stuff. Well, it was mostly that he just spends most of his time on it just trying to smear me and trying to make all these connections that aren't there, and they're basically just lies. And it's like, why would I want to put that up there? But since you complain so badly, I put it there. I put it in here. People can see what you have to say, or what Grok has to say, rather. And yeah, people can refer to his article and then look at what I'm saying, because he puts all of these really bad examples out to disprove my work. Like, for example, there was one example with a human. He said a human adapted strain of I think it was Staphylococcus aureus, and I had shown in this article how bad of an example that was, because it already had a long past with sheep, right? And there was another paper. There was actually another paper on complete genetic remodeling that occurred from a host switch from human to sheep, ruminants, or ruminants, maybe that's goats or whatever, but it's ruminants, whatever it is. I think it's sheep, though. Yeah, I'm pretty sure it is. But it was the same exact thing that he cites, only this paper showing radical genomic remodeling because of it. So his little example was just completely demolished by that other study. And he totally and completely failed to take into account the dynamic that I am talking about. I'm not talking about just simple animal adaptations. We're talking about something much more sophisticated. We're talking about rolling a bunch of tick-borne diseases together and then passaging them, and giving them new vectors and new hosts, all rolled together like that. There is immense potential for things to change, especially when they're released into different ecosystems worldwide. That's where I think a lot of the divergence actually happens, because it's such a dynamic situation. You don't have the set mutation rate until something has already adapted itself, set itself up in an ecosystem, and is thriving. Then once it starts thriving, then it goes back to a slow mutation rate, because it's made all of the necessary rapid jumps, and it's rapidly evolved, and then it starts purifying what it doesn't need. So actually, it's like the reverse. It's like the opposite of selection pressure. At first, when it's released into the ecosystem, it's under massive selection pressure. It starts getting used to all the hundreds of new animals that it's going to be infecting, right? That's where a lot of the divergence happens. It's in the beginning. It rapidly evolves, and then then it starts thriving, and then it purifies all of the things that it doesn't need, and then it's like the opposite of the selection pressure after it's thriving. Okay? And this is a very long article about all of that. And I actually showed at one point that some people thought that a host switching, or so they were talking about Borrelia. Let's see, which one was it? It was a Borrelia in the Lyme clade, right? And it was a scientist saying that he thinks it may have changed into a new species due to a host switch. And then, like Henjen tries to make all of these points on saying you can't change things into new species, and a host switch would not cause that. But here's a guy saying it in the paper where he's saying, I wish I could find it. I can't really find it. I'm using my Kindle here. Oh, so it was a paper, Borrelia bavariensis, vector switch, niche invasion, and geographical spread of a tick-borne bacterial parasite, where he said, throughout its very extensive geographic range in Eurasia, B. bavariensis is associated only with small mammals, implying that a host switch occurred at some time in the past that allowed, or perhaps even was the cause, of the divergence of B. bavariensis or Borrelia bavariensis from Borrelia guarinii. There you go. I mean, he's saying that. Why would he be saying that if that wasn't possible? And he's also assuming that the 1939 strains of Borrelia anserina that Traub and Schaefer had are the exact same profile as they are today. Like they haven't changed one bit. And they can change considerably. And there was actually a study on E. coli, just having it in a lab under certain conditions, and that changed considerably. It was some long-term experiment. But this is a very long article, and it goes over, because another thing that's interesting about this is that when you compare, so Borrelia anserina is closest at the most conserved gene, the 16S gene. It's closest to the Lyme clade out of all the relapsing fevers. Out of all the relapsing fevers, Borrelia anserina is closest to the Lyme clade. But the other thing, and a major factor here, is also that it's basically like a morphological match. It's also very close morphologically. Morphology is like its form. It takes note of length, wavelength, flagella, pointed or blunted ends, a couple other factors. And I actually put the graphs on here showing these other relapsing fevers are considerably different. But when you take Borrelia burgdorferi and you take Borrelia anserina and you put them side, when you compare those, they're very close. Very close. There would be probably indistinguishable. Although some have stated that Borrelia anserina is typically longer than Borrelia burgdorferi. They've also found some Borrelia burgdorferi that are just as long. So it's not really like it's not definitive. It's not something it's not a blanket statement. You can't say that they're all longer, because and I think during that divergence, Borrelia burgdorferi probably made even more adjustments to itself and may have generally gotten shorter in morphology, because those things can happen too. But it's like an exact match to to Borrelia anserina morphologically. And all of the other relapsing fevers are different. So I thought that was really interesting. And I thought it was interesting that, they've been, another really interesting aspect of this is that the Lyme clade of Borrelia are the most dominant form of Borrelia on the globe. Like, they are orders of magnitude more abundant, 10 to 100 times, I would say, more abundant than all of the other Borrelia, the relapsing fever. And they were basically traveling throughout the globe, looking for all the different forms of Borrelia, spirochetes, and they didn't find a single sample of it. They didn't declare a single sample of it until 1982. But yet they're saying it's thousands, hundreds of thousands of years old. And there's a paper talking about how the phylogeography between Borrelia burgdorferi and its tick vector, Ixodes scapularis, is contradictory. There's a paper, it's called Uncoordinated Phylogeography of Borrelia burgdorferi and its tick vector, Ixodes scapularis. It says, despite the intimate association of Borrelia burgdorferi and Ixodes scapularis, the population structure, evolutionary history, and historical biogeography of the pathogen are all contrary to its arthropod vector, the black-legged tick. And so papers are trying to say that it came because of deforestation in the Ixodes tick. But this is basically saying its phylogeography is contrary to this tick. And all the way up to the 1950s, they were saying the black-legged tick has no human diseases whatsoever, right? Then all of a sudden, it's got the same mix of infections that Erich Traub was studying in 1939 under his professor, under his mentor, Carl Beller. He was his assistant professor. He was helping him. So he was helping him also with the when they were doing that book where his mentor published the chapter on heart water and the animal rickettsiosis. You think he didn't read that? Are you kidding me? He was helping him put together. He was helping him. He was learning about all this stuff at that time. He was very well read when it comes to literature. He was very well read when it came to literature, and you could be damn sure he read probably every paper in that book, especially from his mentor. Because, yeah, like Henjen was saying, he probably didn't even read it. Fucking idiot. And, Henjen's only reliance is on the genome. That's the only thing he looks at. He thinks that the genetics, the only thing that matters is the genetic sequence, the genome, and looking at relationships of the genome. But there are many limitations in the genome data. Not just in analysis, but also collecting it. And I put a little section about that, and I talked about, we don't know if this whole 25% divergence, we don't even know if that's really an accurate number. But I did say, for the benefit of the doubt, I'll give him the benefit of the doubt and treat that 25% as though it's real, because the distance is not actually a hurdle to my reconstruction. It's something that was expected. Something was expected. The genetic distance was something that you would actually have expected when he released that thing across the globe. Being that Borrelia anserina didn't have any of the qualities that the Lyme clade had. And then he basically changed that thing from the inside out. He made it cause new disease. He adapted it to new hosts. He gave it a new vector. Um, putting it in, and then putting it in all these different environments. Borrelia anserina was a tropical disease, right? So like Africa, dry, humid parts of Africa and in the Middle East, I think. And then he, gave it completely new atmosphere. I mean, if there was a chance for genetic remodeling, this would be it. This for radical adaptation and evolution, this would be the circumstances. And it's man-made. That's the thing. Science likes to look at all this stuff as though it's like it took millions of years, because they don't want to talk about man-made bioengineering. They don't want to talk about how man has been altering all of this stuff over the years, and how that could actually, a lot of stuff they're saying that is, because they talk about events that may have occurred, and they always backdate it many thousands to millions of years ago, but they really don't know when it happened, right? They're just saying that because they have to cater to this slow evolution model. So it's just dogma. It's this evolution dogma that they have to see things everything has to happen slowly by point mutations. And he also, I pointed out some of the BLAST database limitations in blind spots, because that too also has blind spots. Because he's saying that if it had inherited anything from foreign microbes, he would be able to find a sequence, and he would be able to find the sequence on all of these other microbes. But he doesn't understand that when Borrelia is inheriting other foreign genetic material, it chops it up and recombines it, repackages it. So it's also fragmented, highly fragmented, in highly fragmented form. So you're not going to have these long reads of information. It's going to be very short little bits and pieces that it's so fragmented, you're not even going to be able to tell that it's, from that microbe oftentimes. And especially if we're talking viruses can change considerably from the days from when he was back in his day. If you take the same virus, from 1942, and then you look at it in 2026, it could be radically different than that one. And so there's a lot of different things to take into account here. So many things. And there was actually a case where they found material they thought was from the pox viruses in its main chromosome. And Borrelia burgdorferi has this segmented chromosome that at first there were certain parts of the chromosome they didn't know if it was part of the plasmids or if these were actually part of its genome, like its main chromosome, the core genome. And they eventually found that it was. So at one point they were talking about linear plasmids of Borrelia burgdorferi have a telomeric structure and sequence similar to those of a eukaryotic virus. But then they realized that it was actually these linear plasmids functioned like mini chromosomes. They said these observations and the finding that the Borrelia chromosome migrates in pulse field electrophoresis gels as a linear molecule of 950 kb, I forget what that stands for, but raised the possibility that the linear plasmids are actually mini chromosomes. And then later on, you can see that it's part of the main chromosome that makes up its segmented genome. And that was published in Telomeres of the Linear Chromosomes of Lyme disease spirochetes' nucleotide sequence and possible exchange with linear plasmid telomeres. There's like this exchange going on. They found they have these hairpin ends that resembled pox viruses. And there was another paper that talked, these observations suggest there has been historical exchange of genetic information between the linear plasmids and the right end of the linear chromosome. So there's a lot going on with Borrelia burgdorferi. It's a very complex, it's a complex bacterial genome, right? And that's another thing about the whole genome sequencing and the genome data, is that there was actually, it was an article I found in my inbox. It was an article, I think it was from The Scientist. It said short read sequencing makes it impossible to fully resolve complex microbial genomes. And all of Henjen's criticism of my work is based on this fragile data alone. But yeah, I mean, he totally missed this thing about the pox virus. He was saying if it had any material from anything else, I would have been able to spot it in BLAST. And he didn't pick this up. And I don't think they used BLAST when they created this paper, because they showed the different you can't see that. Never mind. They showed the sequences and how there's the viruses had a 67% similarity to it. And like, I really go deep into this, pointing out all of, like, just totally showing how all of his debunking is complete nonsense. Like, he's not taking so many things into account, and he's not understanding what I'm actually saying. So I made this much longer one to clarify that I'm not saying the distance even happened right in the lab. And I never said that any of those things I listed in my last article, each on their own, were responsible for the 25% divergence. I never said that. I'm saying all of those things I talked about in my first paper were very likely to have been responsible for the divergence in that 16S gene. That's what I was more focused on. But as far as the larger 25% divergence, yeah, that would have been expected for something like this. The way he completely remodeled it, that would have been expected. Absolutely. I mean, it would be totally unheard of if it was completely still the same, because it's a totally different germ at this point. It has been completely changed by its host, its disease, its tissue tropism. So Borrelia anserina basically just floods the bloodstream. It doesn't bury itself deep into the tissues and brain like the Lyme disease spirochetes do. And that's another thing. So it's been changed in every way. Of course it's going to be radically different in its genetic distance. It's going to rapidly evolve and be that way. And there was another example. I talked about the fact that also when you have a whole mix of infections that induce this immune deficiency or immune tolerance, you're going to get this, you're going to raise the mutation rate way up. And especially since it's in that mix of infections, that's a dynamic that I don't think really any papers that are around today have studied stuff like that to the level that this was at. Um, I mean, they showed that people with immunosuppression, showing the SARS coronavirus 2, during the COVID-19 pandemic, it was found that one immunosuppressed person infected with SARS coronavirus 2 had 20 mutations in a single infection. And while viruses have a higher mutation rate, both still have a much higher mutation rate in the immunosuppressed. And there was a case involving syphilis in an immunosuppressed syphilis patient co-infected with HIV, spreading the disease around. And there was this YouTube video where it's talking about how two types of the syphilis spirochete combined after genetic recombination and underwent intrahost evolution, bringing confirmation that bacteria don't always change slowly. And the guy said, we have documentation going back decades that this particular bacteria changes slowly. But we've now been able to directly observe that different strains can and will recombine genetically to create new strains. It's not to say that this has never happened before. It's just that now we have evidence of seeing it firsthand. So that's important, because even with things that are thought to have a very slow mutation rate, well, here's a situation where it made this massive leap, where it underwent intrahost evolution and recombined, and two different types combined. And I also think it's very possible that Erich Traub may have incorporated a bunch of different strains of relapsing fever Borrelia in with this as well in his adaptation to try to give it more fitness. I mean, I don't know exactly what he did. I'm going on limited data, but I have enough of it that really starts to explain the picture. And I also talked in this article about his expertise on virus and bacteria interactions. Because he talked about how viruses could act like bacterial genes in some ways, or the prophages could act like prophages are bacterial viruses. And he talked about how they could integrate into the genome of the bacteria and act somewhat like bacterial genes, and also undergo recombination and things of that nature. So he definitely had the expertise, and they were already studying bacteriophages and all of this very complex stuff at Ensel Reams. They were already doing that, and they were already working with mycoplasma, which also acts as gene shuttles. It's bacteriophage-like. It's not the same as a bacteriophage, but it can act bacteriophage-like. It has phage-like properties and acts as a gene shuttle between the different infections. And I mean, I just really laid it out in here. I'm not going to go over this whole thing because it would be a really long video. But I just kind of wanted to point out that it's up there. You can read through it. And this has like 177 or this has like 177 citations or something like that. And there's a few pictures, showing some of Erich Traub's adaptation work. Some of the mycoplasma images from 1947. And I think it was from work during the war. I think it was just published in 1947. But I really laid it out here. And I'm happy the way this came out. I mean, I can understand why it seemed like such a stretch to him at first. Especially not being... He doesn't really know anything about all this stuff. He doesn't know anything. He has to ask Grok all of this. And Grok is terrible. I mean, not Grok itself, but the AIs are terrible. They'll tell you... they'll basically cover for the establishment, and they are drawing off limited data. Like, so I was able to convince Grok when it had, I started telling him about Henjen's position and what Grok was telling him, and then I tried to lay it out to it what I thought and said, no, 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 this is not possible, it's not possible, like it was telling him. But then after I started going over like everything I knew, and it started to slowly see like, oh, you know what, you might be right about this, actually, you might be right about this. And now it thinks that my reconstruction is actually the most plausible, and it explains a lot of the anomalies surrounding the Lyme disease, the Lyme clade, and how, all of the anomalies, like why didn't they find a single sample of it before 1982 if it's the most dominant form of Borrelia on the planet, you know? And there's a lot of contradictions in the whole slow evolution theory of Lyme disease as a being a natural thing. And I talked about how they tried saying it was in a mummy, right? Or they tried saying they found it in a mummy from thousands, or an Iceman or something from thousands of years ago. But they didn't even know what bacteria it even was. When you go and you actually read the paper, they don't even know what bacteria it is. They can't even say if it's even the genus Borrelia. They don't even know that. They say there's a 60% chance. Like, well, what does that mean? So it's 50-50, almost 50-50? Like that's not science to me. And then they have these other instances, like I talked about in my book, where they find, they take samples of mouse tissues from the 1800s, or ticks samples from the 1800s, and they don't even test for the 16S gene. They don't even test for the flagellin sequence. They only test for OspA, which is extracellular DNA, and that would not make sense to do that, because extracellular DNA changes regularly and rapidly. And even Marconi put a lot of papers out on how variable that OspA thing is. And you can have stuff that cross reacts with it. There's all sorts of stuff that can happen, and that's why you have to try to test for those conserved genes like flagellin. And they didn't do that in those older ones, but they did that for the studies with ticks in the 1950s. So it's very inconsistent, because they're trying to create propaganda. They're trying to sell a narrative that's not really there. They didn't find it thousands of years ago, and there's papers saying that oh, it's been here for thousands of years. That's all theory. That's all theory. That's their theory. It's not fact. They state it as though it's fact, but it's all theory. Based on phylogeography, they made the claims that it's been here for thousands of years, and they have no idea of knowing. They would have no way. They have no proof of that. It's all theoretical, and it's really poor science, in my opinion. But I covered some of that in here. I just really laid out how I talked about how different ecosystems can change microbes considerably, and there's a lot of published work that would substantiate that, right? So go ahead and check this out. I'm so glad that I could finally bring this to you. I've been working on this for months, because this is something that I had to take a lot of time to sift through this. Like, I have been writing this for months, actually. Like, I really wanted to try and lay this out, but it had to be done carefully and slowly. And I don't, he just uses Grok and asks Grok and copies and pastes what Grok says in all of his work to try to debunk me. I sit here and I actually research. I sit here and I actually go through everything. That's why it takes so long to to respond. I'm not going to sit there and argue back and forth with this person who I don't even know who it is in a comment section. That's just absolute dross. Like that's what somebody who you would consider complete dross would do. I just don't have time for it. And then, he will, I had unblocked him at one point because he was starting to be somewhat civil. And then I went to bed and I wake up to like 100 comments of just trying to smear me and stuff. And so then he wonders why he gets blocked. It's like, dude, I'm not going to deal with stalking and harassment. You're not going to sit here and stalk and just totally overwhelm all my posts with these shit comments. But don't take that as I'm afraid to engage with your information. Because that's not at all the case here. I did respond. This is my response. And I'm sure he's going to take this to Grok, and Grok will come up with all of these fancy replies as to why I'm wrong, but each of them will be complete shit just like his last paper. So go fix up your stupid little fucking document, and we'll talk soon, Henjen. And for everybody else, check out my Substack article and share it widely. Feel free to reach out. And also, I did a interview on Sherry Tenpenny today, so I'll have a link to that at some point. And take care of yourselves, be yourself, build yourself, and I will see you on the other side.